Okay, great. Thank you. So yeah, it's my pleasure to start session 2, which is celebrating 40 years. Yes, 40 exclamation mark. The embellishment's mine, not Martin's. 40 years of the Smith Lab with talks and comments by distinguished alumni. As Martin said, I'll be co-chairing this session with David Eastman, who's in the room, and David is from University of California, Riverside. We'll get straight into it because we're behind time here, but our first topic is MPTP, paraquat, and Parkinson's disease. And our first speaker will be Dr. Donato De Monte, with comments by these alumni, which will include Doctors Martha Sandy, Gunilla Ekstrom, and Sarah Jewell. Now, Martin did send me a photo that I couldn't resist using to introduce our first speaker, Dr. De Monte. I've known Dr. De Monte for about 40 years now. Here you see him in true action mode in the 80s. It was pretty impressive. Now, I haven't followed his football career, but his soccer career, I should say, but I've certainly followed his scientific career, which is incredibly impressive. And so I'd introduce Dino De Monte, who is from the German Center for Neurodegenerative Diseases. Dino, take it away.
Okay, can we switch
to... can you hear me?
We can, Dino. We need to... I'm just
going to quit. Video is telling me that I cannot start the video because the host has stopped it.
Okay.
I don't know who is this car. Okay, start my video. Okay, let me see.
Can we put Dino to full screen now?
Okay, I think now you can see the video. Okay, let me start sharing the screen and see if you can see it. I don't think I can.
Have we got the security allowing sharing?
Share. Okay. Can you see it?
Yes, Dino. That's great. Thank you.
Excellent. Okay. Well, first of all, I would like to. I'm really sorry for not being there with all of you, and of course, one of the reasons will be to have an opportunity to meet and see again after a long period of time many people with whom I have shared incredible experience at the beginning of my career. I also would like to thank Martin for the invitation and this opportunity to give a short talk, and I also want to thank him for the title of my talk, which is MPTP, Paraquat, and Parkinson's disease, which pretty much could have been translated into MPTP, Paraquat, Parkinson's disease, and the meaning of life. And he appropriately gave me 10-15 minutes to talk about this. So thank you, Martin, for that. But I take the challenge. So I will start and I will give you, and this is of course for you, most of you perhaps shifting gears in terms of the topic, but I will give a general quick introduction on Parkinson's disease and then focus mostly on paraquat and Parkinson's disease, the potential relationship between paraquat and Parkinson's disease from the experimental standpoint, which is what we have investigated over the years. So first slide, this is just like a summary of milestones in the Parkinson's disease research, starting from 1817, the first description of the disease. In 1912, Frederick Louis described, these are the designs.
Dino, your slides are not advancing.
Okay.
We're still on the title slide.
Ooh, okay, let me try again. Okay. Can you see now?
Yeah, there you go.
Okay, let me try again. Okay, can you see it?
Yeah, we can see it. Yeah, the slide's good.
Okay, can you see it?
Sorry, yeah, the slides are good now. It's fine.
Okay, good, thank you. So as I was mentioning, 1912, the description of these intraneuronal inclusions that taking the name of Frederick Lewy are called Lewy bodies. And just to emphasize, every patient with Parkinson's disease, when their brain is analyzed histopathologically post-mortem, has these typical inclusions within the neurons. Then in the 60s, Arvid Carlsson, who actually won the Nobel Prize in 2000, made a major breakthrough with identifying dopamine as a major neurotransmitter involved in Parkinson's diseases. And as a consequence of that, as a corollary to that discovery, it was also discovered that an area of the brain called the substantia nigra is a key area being involved in Parkinson's disease, and the degeneration of neurons in the substantia nigra are a key pathogenetic process in PD. Then we jump to 1997, and in the lab of Bob Nussbaum at NIH, this important discovery was made first from through genetic studies. He identified alpha-synuclein and mutations in alpha-synuclein as the factor associated with Parkinsonism in families with familial Parkinson's disease. The other interesting thing, then it turned out that alpha-synuclein, this at the time was a known protein, but now very known protein, is alpha-synuclein, is a major component of the Lewy bodies that Frederick Lewy described almost a century before. Now you can look at this graphic and you can say, well, did anything else happen between the 60s and almost at the end of the century? And I think something very important, and think this gap, this important gap, was filled in the middle of the 80s. And this is when I happened, and I was in the lab with Martin, and with him we started working on MPTP and paraquat. Actually, initially we were focusing on paraquat, and then the MPTP came out. And because of the chemical similarity between MPTP and paraquat, we became involved in this study. So this is how it all began, basically. We started investigating and comparing the toxic effects of paraquat and MPP+, which is the toxic metabolite of MPTP. And it was a very prolific time and you know, I'm summarizing here at the bottom some of the publications that we made, but there are quite a few more that I am not listing. But the bottom line of these studies was that this comparison clearly showed that MPP+ and paraquat are not very similar. In fact, as you see on the left, on the graph on the left, paraquat is a classic oxidative stress-inducing compound. MPP+ is not particularly good in inducing.
Sorry, Dino, your slides are not advancing.
Okay, that's so. I will maybe I'll just shift to showing. Okay, can you see them now?
Yes, yeah, we can see it. Yeah, great.
Okay, so as you can see on the left, oxidative stress is involved in paraquat toxicity, and whereas with MPP+, it was very clear that the major target of toxicity of MPP+ and MPTP were mitochondria, and this led to a loss of ATP, for example, when we were incubating cells in the presence of MPP+. So this was the work that we did, and I participated in Martin's lab, and I thought that pretty much the story was finished, was concluded, and the answer was the two compounds are different, and most likely paraquat has nothing to do with Parkinson's disease. Needless to know, a few years later, I had to change my mind. Can you see the slides?
Oh, yeah.
Okay, great. And so almost 15 years later, the debate about paraquat and NPP plus and NPTP continued. So I decided somehow to do the key experiment that would have probably put the debate to sleep. As I mentioned before, in Parkinson's disease, the degeneration of neurons in the substantia nigra are a key pathological feature of Parkinson's disease. So I thought, well, maybe we should inject paraquat to experimental animals and see if paraquat was also able to cause the loss of neurons in this area of the brain. And that's exactly what we did, and this is the results, some of the results of the experiments that we obtained. We injected paraquat at different doses, and as you can see in the graph, this is the number of neurons in the substantia nigra, and in a dose-dependent fashion, paraquat indeed caused neurodegeneration of these neurons. We went on and did additional studies in which we also clearly identify a relationship between oxidative stress, neurodegeneration, and exposure to paraquat. This is an example of tissue of the substantia nigra that is stained for nitrotyrosine, which of course would identify protein that will be nitrated as a consequence of oxidative stress. As you can see with paraquat, there is a significant nitration of protein in the cells in the substantia nigra. The other thing that we have been doing, which I think is very important, and I won't go too much into details, but we also think that an important component in this neurodegeneration oxidative stress caused by paraquat is microglia activation. Microglia are the key inflammatory cells in the brain, and when there is an injury, they're activated, and their activation itself can contribute to oxidative stress and pathological processes in some instances. That seems to be the case with paraquat. So yes, paraquat causes neurodegeneration through oxidative stress and possibly microglia activation. Now let me go back to the other screen and let me show the next slide. So once again, I thought that the story was ended, was pretty much done for me. But 15 years later, once again, the paraquat story continued. And in this case, the relationship that we investigated was the relationship between paraquat and alpha-synuclein. As I mentioned before, alpha-synuclein is a key pathological protein involved in the pathogenesis of Parkinson's disease. These are Lewy bodies, the Lewy inclusions that I've also mentioned before, that are stained specifically for alpha-synuclein. As you can see, alpha-synuclein is indeed a major component of Lewy bodies. So we asked the question of whether or not paraquat was also capable of affecting somehow alpha-synuclein pathology. And when we asked that question, I want to also make a premise that the two key mechanisms that are probably involved in alpha-synuclein pathology is protein aggregation, so the tendency of alpha-synuclein to aggregate, and perhaps this aggregation is directly involved to the formation of inclusions in the neurons, and also the tendency of alpha-synuclein to jump from neuron to neuron, and through this mechanism to spread throughout the brain. So we investigated the relationship between paraquat and alpha-synuclein. The first thing we did was we looked at oxidative stress in cells that expressed alpha-synuclein. And what this graph shows that oxidative stress is significantly increased in cells that express alpha-synuclein. Alpha-synuclein is in red, and oxidative stress is indicated by those dotted points that you see is the oxidation of the dehydroetidium. So indeed, paraquat has a significant effect on alpha-synuclein, on oxidative stress in cells that express alpha-synuclein. I mentioned aggregation, and we looked at the effect of paraquat on alpha-synuclein aggregation, and just in the graph that quantify aggregated forms of alpha-synuclein measured using proximity ligation assay, clearly you see that in the presence of paraquat, alpha-synuclein aggregation becomes significantly more pronounced. Finally, I also talked about spreading of the protein from one region to another, and to make a long story short, the graph shows that in the presence of paraquat, alpha-synuclein also become significantly more prone to move from neuron to neuron and from brain region to brain region. So clearly, oxidative stress, aggregation, and spreading of alpha-synuclein are significantly affected by paraquat. So let me now go back to the next thing. And what I wanted to show you is that, of course, I don't have time to draw a lot of conclusions about paraquat, Parkinson's, and so on. And I think you probably are in a better position than I am in drawing some conclusions in terms of the potential involvement of paraquat as a risk factor for Parkinson's disease. But what I wanted to focus on is something that we discovered is rather an interesting new finding, and that is the effect of paraquat on aging. And I have a really personal experience on it. Here on the left, you can see a picture that I took when I was actually working with marketing in Berkeley, and I was commuting with my little scooter from our apartment in Oakland and go every day to the lab in Berkeley. And just to show how clearly this effect of accelerating aging by paraquat is evident, this is what happens after a few years of working with paraquat. Now you can also say, well, we all scientists, this is an N equal 1. How can you draw conclusions based on experience, as much as, you know, traumatic experience by yourself, but experience of N equal 1. So I thought just a bit about that, and then I realized that I do also have the perfect control, and so I went and I clearly went back and I find a picture of Martin, and you can see the accelerated aging on top and more successful aging at the bottom. And of course, the big difference here, the major difference is my exposure throughout my scientific career to paraquat compared to the little or very little exposure, no exposure of Martin to paraquat. So in conclusion, I would like, of course, to thank Martin for giving me the opportunity to initiate the work with paraquat and stepping out of it and leaving that completely to me, which of course was important for my career. But actually, I have many reasons really to thank Martin. Martin gave me to jumpstart my career in his lab. He gave me the opportunity to work in a fantastic research environment with wonderful colleagues. Some of them are there, Martha Gunilla, of course, David Eastmond, and they were all in the lab when I was there working with Martin. Of course, he has taught me quite a lot of things in terms also, for example, managerial skills. Other people before me have emphasized that, and I did appreciate the ability of Martin to step in at the right time and to stay aside when he thought it was better to let us go and do whatever we wanted to do. I think he had this incredible ability to be able to find the right time to step in or to step aside. And of course, for me, the most important thing is to thank Martin for having been also my friend for so many years. How do you recognize a good friend? Well, one of a good sign of friendship is when people get in touch regularly. And of course, you friends can get in touch on a birthday, on an anniversary, on special occasion. Well, with Martin has been very special because I could count on him to get in touch with me when there was a soccer game between England and Italy. And of course, in particular, when he thought that England had a chance or a greater chance to beat Italy, I was regularly receiving emails and getting in touch with Martin. So I thank again Martin for his also being a good friend with me, and unfortunately, is getting in touch with me, has not been most of the time successful with the English team. And this is just a little example of one of the latest interactions that I had with Martin on this topic. Again, thank you, Martin, very much. And thank you to all of you for having listened to my presentation. Thank you.
Thank you, Dino, I think. Yeah, that was very good. I just, before we take a break, Martha, do you want to come up and give comments? Or what was your plan with Dino and Gunilla?
I think Gunilla and I will.
Okay. So Gunilla Ekstrom, are you on from Sweden? She was in the lab with
Yes.
Dino.